Hello future researchers! Hello, kumusta kayo? Welcome back to my YouTube channel. This is me again, Teacher May. Syempre, ang kasama mo na naman sa iyong research journey. If you're new to this channel, don't forget to subscribe, like, and hit the notification bell for more updates in research. Science can be both entertaining and risky at the same time. You can do a lot of harm if you don't know how to use chemical and laboratory equipment properly. Though curiosity is a good thing, you should always have all the necessary information about lab chemicals before using them. It can be difficult and dangerous to conduct experiments without having accurate information or experience. How can you possibly imagine going in the right direction if you have no idea what chemicals are used, for what purpose, or how to handle this necessary equipment, especially during experiment? Yes, it is important to use the appropriate laboratory equipment. You will not succeed in your mission unless you conduct an experiment first. Apart from the performing the task without prior experience can be risky. You wouldn't be able to figure out how to fix your errors. Science is undeniably important to us, but going without knowledge can be a potentially dangerous situation. When two chemicals are mixed without understanding their properties, a blast will occur, potentially killing you. It would be very dangerous not just for you, but also for the others. For today's lesson vlog, what we're going to discuss is the proper handling and setting of laboratory equipment. This is for the Module 6 in Week 6 of Quarter 3 in Research 2. For the content standard, the learner demonstrates understanding on the principles and types of research design, sampling types and methods, and appropriate laboratory techniques and methods necessary in conducting a science investigation. For the performance task, the learner should be able to make a draft research design in his or her data logbook, including protocols, methods, and procedures. And for the learning competency, the learner should be able to apply proper handling and setting of laboratory equipment. Let's start with the Biological Safety Cabinets, or the BSC. As you can recall your topic when you were in grade 7, it has been discussed under the BSL4 that there are two types of cabinet. The first one is the laboratory cabinet. This is the biological safety cabinet, and the second one is the suit laboratory. Biological safety cabinets, or the BSC. Biological safety cabinets should be used when working with pathogens, tissue culture, and human samples. So what do we mean by pathogens? We know that pathogens are those bacteria that are causing diseases. Biological safety cabinets, these are primary means of containment developed for working safely with infectious microorganisms. So how to prepare in using this biosafety level cabinet? Turn on the fluorescent lights. Check air grills for obstructions. Switch on blower. Allow air to purge workspace for 5 minutes. And of course, we have the disinfection. Spray or swab all the interior surfaces with appropriate disinfectant or the germicide. So it's very important to clean all the necessary parts of these cabinets. How do we assemble material using these biosafety cabinets? First, pre-plan and have all supplies ready for the entire procedure. Introduce only material required to perform procedure, and do not block air intake or exhaust grill by overfilling the cabinet. Place material such that clean and contaminated items do not meet. Place contaminated material container at right rear. Okay, so before you use this, allow air purge period with no activity inside. So you have to leave the blower on. So as you can see in this picture, this is the airflow scheme. And this red arrows, okay, this is where the contaminated air flows. And we have the ambient air, this is the green one. And the blue one, these are the clean or the filtered air. Okay, you have to record the static pressure on the appropriate paper. Yes. So it's really important that you record everything that you do here, especially just like this static pressure. For the personal procedures, do not forget to use protective clothing such as the gloves, the mask, etc. Okay, as appropriate, just like this. You have also the face shield and the PPE. In performing the procedures, introduce hands into workspace. Work carefully and methodically, for example, from clean to work area to discard. So it's really important that you are working in a very clean area to avoid contamination. Do not remove hands from workspace until procedures are complete and all critical material is secured. Remove gloves into contaminated material. Yes, again, remove gloves into contaminated material container. And for the post-use, so after you use the biosafety level cabinet, you have to allow air purge period with no activity inside. So you have to leave the blower on. So actually class, so this biosafety level cabinet, these are used by the experts or by the qualified scientists. Yun talaga mga nagtatrabaho na sa mga laboratory institutions. So for the terminal disinfection, use fresh gloves, remove materials to incubator, to biohazard bag, autoclave as appropriate. Spray or swab all interior surfaces with appropriate disinfectant. Yan. Okay? As you can see here, this is the blower. So before, of course, you have to turn this on. Okay, but after, so you have to shut down and turn off the blower. Replenish communal supplies. So in replenishing the communal supplies, you have to refill the bottle of disinfectant. You have to put a new plastic bag in the waste bucket. Other helpful hints with biosafety cabinets: you have to keep activity in the area surrounding the hood to a minimum to reduce the amount of air disruption in the hood. Place everything at least 4 inches inside the work area to reduce chance of contamination. So this is the biosafety cabinet. So you are doing the experiments inside this cabinet. So the virus and the bacteria will be confined in this area. If a spill occurs, clean it up immediately before continuing work. Do not use an open flame in a biosafety cabinet. Every time the cabinet is used, record the static pressure on the chart provided by EHS. What to do in case of a cabinet failure while working? Seal containers, surface, decontaminate, and remove any biohazardous material. Decontaminate the interior of the biosafety cabinet. Switch off the power if the motor is making noise. Place a sign on the cabinet to indicate that it is broken and must not be used. If personnel may have been exposed to infectious material due to cabinet failure, then the supervisor must be promptly notified and an incident report completed and appropriate first aid and medical follow-up action taken. Clean air benches are not biological safety cabinets and should never be used with pathogens and other materials that are potential aerosol hazards. These benches also protect the product, not the user. This is an autoclave. An autoclaves are used to sterilize items potentially exposed to biohazards. Autoclaves operate with steam and pressure. Both of these can be dangerous, so autoclaves should be used with caution. When loading an autoclave, do not overload by stacking or overflowing containers. Some general guidelines as follows. For liquid, containers should not be any more than 2/3 full. Put in an autoclave safe container. Be careful when removing to prevent bumping or liquid boiling over. For solid waste bags should be open so that steam can penetrate to sterilize all items. Each cycle should be about 1 hour. Now, let's move on to important safety practices for autoclaves. Load the autoclave properly per the manufacturer recommendations. Before loading containers of liquids into the autoclave, the caps must be loosened. Use an autoclave safe tray with a solid bottom and walls to contain the bottles and catch spills. Don't load plastic materials that are not compatible with the autoclave. Individual glassware pieces should be within a heat-resistant plastic tray on a shelf or rack and never placed directly on the autoclave button. Make sure the door of the autoclave is fully closed and the correct cycle has been selected before starting the cycle. Wear heat-resistant gloves when cracking the autoclave door open after a run. And remember, before removing autoclave items, wait 5 minutes for loads containing only dry glassware and 10 minutes for autoclave liquid loads. When removing items from the autoclave, use heat-resistant mitts. Be alert for autoclave liquid bottles still bubbling. Let liquid loads stand in an out-of-the-way place for a full hour before touching with ungloved hands. Hot glassware and scalding liquids will cause burns and serious harm. Do not autoclave items containing bleach or phenol. And this is a PPE, which is the personal protective equipment. And this is what must be worn when you work in the laboratory. You have the eye protection, the lab coat, the long pants, closed-toed shoes and rocks, which is no exposed skin around the feet, lab gloves when required. So PPE including long pants, closed-toe shoes, lab coat, safety glasses, and heat-resistant gloves should be worn when loading and unloading an autoclave. An SOP for using an autoclave is available on the AFNS Safety Drive. So, understanding autoclave, there is a standard operating procedure. Now, let us proceed with a centrifuge. So, remember that this centrifuge is the device that is being used in the research method, which is centrifugation. So, this is all about that this is used when you separate mixtures through spinning. A centrifuge is a device that uses centrifugal force to separate various components of a liquid. This is achieved by spinning the fluid at high speed within a container, thereby separating fluids of different densities, for example, cream from milk or liquids from solids. A centrifuge is a high-speed spins generated by centrifuge can generate or can create large amounts of aerosol if a spill, leak, or tube breakage occurs. An SOP for using centrifuges is available on the AFNS safety drive. To avoid contaminating your centrifuge, you have to check glass and plastic centrifuge tubes for stress lines, hairline cracks, and chip rims before use. Use unbreakable tubes whenever possible. Avoid filling tubes to the RIM. Use caps or stoppers on centrifuge tubes. Use sealed centrifuge buckets or rotors, which can be loaded and unloaded in a biological safety cabinet. Decontaminate the outside of the cups or buckets before and after centrifugation. Inspect O-rings regularly and replace if cracked or dry. And of course, make sure to ensure that the centrifuge is properly balanced. Do not open the lid during or immediately after operation. Attempt to stop a spinning rotor by hand or with an object or interfere with the interlock safety device. Decant supernatants carefully and avoid vigorous shaking when resuspending packed cells. Okay, syempre, clean spills promptly.
And now, let us move on with lyophilizers. This is a lyophilizers, and this is also known as the freeze dryers. So these lyophilizers usually is being used when it comes to research method about drying. Aerosols may be produced during operation of a freeze dryer and when material is being removed from the chamber. When lyophilizing biohazardous materials, so first, you have to load the samples in a biological safety cabinet. First, load samples in a biological safety cabinet. Check glass vacuum containers for nicks and scratches. Use only glassware that was designed for high vacuum use. Use a disinfectant containing chop for the vacuum pump exhaust. And after completion of the run, decontaminate all accessible surfaces. And we also have the needles and syringes. Needles and syringes present hazards of spill, auto-inoculation, and aerosol generation and should be used only when absolutely necessary. When working with syringes and needles, the following precautions are recommended. First, perform all operations with infectious material in a biological safety cabinet. So these are the different examples and the different types of biological safety cabinet that we can see in a laboratory setting. Keep syringes and sharps pointed away from the worker and others. Fill syringes carefully. Avoid frothing or introduction of air bubbles. Use luer lock needles and syringes or units in which needles are integral to syringes. Do not bend, shear by hand, or recap needles. Do not disassemble a needle syringe assembly. Place used needles and syringes in puncture-resistant containers. And for the pipettes, selection of a mechanical pipetting aid is very important. Improper handling of pipettes can lead to contamination of the user and/or to generation of hazardous aerosols. Mechanical pipetting aids should be used for all pipetting procedures, never pipette by mouth. Selection of a pipetting device should be based upon intended use, ease of handling, delivery accuracy, user preference, quality of seal formed with pipettes to be used. Liquid should not leak from the pipette tip, whether the pipetting aid can be sterilized. Safe use of pipettes. If infectious aerosols are likely to be generated, pipette in a biological safety cabinet. Handling pipettes as described below will reduce splashing and aerosolization. Plug pipettes with cotton. Check pipettes before using cracked or chipped suction ends may damage the seals of the pipetting aid. Keep pipettes upright while in use and between steps of a procedure to prevent contamination of the mechanical aid. Gently expel contents close to the surface of a liquid or allow to flow down the side of the container. Avoid mixing fluids by alternate suction and blowing or by bubbling air from the pipette. Avoid first full ejection of the contents. Use the short for to deliver, also referred to as mark to mark rather than to contain pipettes, as the last drop of fluid does not have to be expelled with pipettes. Use easier to handle shorter pipettes when working inside the biological safety cabinet. Submerge use non-disposable pipettes horizontally in disinfectant solution. Dropping them in vertically may force out any liquid remaining in the pipette. Now, for the miscellaneous equipment, we have the microscopes. I know you are very familiar with the microscopes. Disinfect the stage eyepieces, knobs, and any other contaminated parts. Select disinfectant that will be effective on the pathogens and non-corrosive to the microscope. And for the water baths, a laboratory water bath is used to heat samples in the lab. Some applications include maintaining cell lines or heating flammable chemicals that might combust if exposed to open flame. A bath or a water bath generally consists of a heating unit, a stainless steel chamber that holds the water and samples, and a control interface. Different types of water baths offer additional functionality, such as circulating water bath that keep a more even temperature, or a shaking water bath that keeps the samples in motion while they are heated. Clean regularly. Add disinfectant, such as phenolic detergent, to the water. Avoid using sodium acid to prevent growth of microorganisms, because sodium acid forms explosive compounds with some metals. Raise the temperature to 90 degrees Celsius or higher for 30 minutes once a week for the contamination purposes. To prevent electrical shocks, unplug the unit before filling or emptying, and have the continuity to ground check on a regular basis. However, for the tissue grinders, homogenizers, or sonicators, so this is a tissue grinders, can also be the homogenizers or sonicators. Allow aerosols to settle prior to opening vessels. Use this in a biological safety cabinet when working with biohazardous material. Wear gloves and hearing protection. For microbiological transfer loops, this is used in aseptic technique, as you can remember. To eliminate the spattering and aerosolization associated with flaming of loops, char the material before fully inserting the loop into the flame. For example, before flaming, hold the loop close to but not into the flame. Alternatively, use disposable loops or a micro incinerator.
This is the end of our lesson vlog in research. I hope you learned something from this vlog. I hope you stay tuned on this channel. This is me again, Teacher Tin May. If you're new to this channel, don't forget to subscribe, like, and hit the notification bell for more updates in research. Bye! See you on my next vlog.